Okay, we'll start in 2 minutes, okay? I think a quick recap will be good. That's what I was requested to do. Yes? Everybody's good with that? Yes? Okay, great. Great. So we'll start right at 10, please. Hey, please be in your cell and it's 1 more minute. Let's see how we can quickly recap this section. Okay, it's 10, guys. So what I'm going to do, I'm going to quickly recap this session. Before I hit record, please see that this session is only for Middletown students because it's a last minute request and I don't want to share recording with other campus students and stress them a day before the test, right? Few hours before the test. This request didn't come from them, just clarifying, but I'll tell your class rep too. And their course director need to decide if they need it. I will share with them. Is that clear, everyone? Please? Right? I don't want to do a last minute thing for anyone. Okay, so
Recording
let's
in
start
progress.
this. So please see, I hope I am recording. I think I am, hopefully. Okay, great. So please see that first of all help files are available on Canvas. Can you hear me well? Can you see the PowerPoint? Everyone? Yes, all help files are available on the Canvas. Dr. Lund from Montana has posted a practice question. Very good. More questions, better. Please do all the questions. They always help you bring back focus. How many lectures you had? You had 4 lectures. As you know, population genetics and tools are itself a session, a lecture in a session. Please do them well. What techniques you had? You had DNA sequencing, microarray, miRNA, and whole genome, whole exome sequencing. How are you going to address this technique? Because remember, I'm going to do a quick recap. My intention is not to teach you here, so that you can quickly refresh the whole section and also refer them. So please see what you need to know about these techniques, principle, advantage, disadvantage, and how you can read them. Did I ask you to know what is sense strand, template strand, and mRNA? So mRNA is same, sequence is same as sense strand or template? mRNA sequence is same as sense or template? mRNA sequence. Anyone? It's same as sense strand, except instead of T, there is U. You have this background knowledge from fall, right? Keep that in your mind. Secondly, what is this technique? As I told you, you have 4 techniques. You have DNA sequencing, microarray, miRNA, and whole genome versus whole exome sequencing. We have clearly identified what we do in DNA sequencing. For example, you want to find out the sequence of this strand. This acts as a template for the newly synthesized strand. Newly synthesized strand is the one which is on the gel. Please know that. It's the newly synthesized strand which is run on the gel. You find out its sequence. Initially, you used 4 tubes, ATGC. This is great in capillary electrophoresis. All you're doing is labeling each nucleotide with a fluorochrome. So what is the nucleotide? What is which nucleotide is labeled blue here? Because this is the newly synthesized sequence and this is the control. Which nucleotide here is labeled blue? C, great. So yes, this is the newly synthesized strand. Please read the question as it is. Yes, C is labeled blue. Please first always decode the newly synthesized sequence, which is run on the gel. Please read as in the question. They are saying this is the newly synthesized sequence. Find out the sequence of the strand that acted as a template for it. Simple. Please always find out complementary first, then read from 5' direction so you don't make a mistake. So the key concept is you should know what are they giving. They have given a newly synthesized strand which was run in the gel. You had to decode it. Here it is reflected in chromatogram. They are simply asking you the sequence of the strand that acted as a template was a template of it. Finish. Find out complementary, read from 5'. I'm just bringing your attention to how to approach this since I'm not teaching but the pointers. You know capillary electrophoresis and each nucleotide labeled with fluorochrome helps you with the initially 4 tubes used, which is ATGC. You have to use 4 different tubes. What is this? This is not X chromosome. This is a chromatogram again where nucleotides are labeled with each nucleotide is labeled with fluorochrome. Fluorochrome. Do you need to know in this exam 2 section some of the inheritance pattern disease? One thing I also want you to bring to your attention is sometimes you may have a disease, or sometimes you may have inheritance pattern mentioned, and sometimes you may have a presentation. So at least those which are in the lectures, be familiar of these presentations. They are not saying it is an inherited PDH deficiency. They are giving you presentation, but you know it's an inherited PDH deficiency. This is an X-linked disorder, especially PDH1 E1 deficiency. You know that in a X-linked recessive disorder, who's a carrier, mom or a dad? Mom or a dad? Who's a carrier, mom or a dad? X-linked recessive disorders. Mom, that's why, how many peaks are you seeing here of carrier mom? 2. 2, very good. So you need to know how to read this chromatogram. Is there any male-to-male transmission in X-linked recessive disorder? Is there any male-to-male transmission in X-linked recessive disorder? No. So the mutant nucleotide peak is G. So this is how you need to, yes, do you need to know the principle, the advantage, disadvantage, and how to read them? Absolutely. Now, as I told you, somebody asked me, what if it was a chromatogram for an autosomal recessive disorder? If I ask you, this is an chromatogram of an X-linked recessive disorder, this may be 2 phenotypically normal couple. Let's assume now we are asking autosomal recessive disorder and 2 phenotypically normal couple. So tell me, will you see 2 peaks in that too if it was an autosomal recessive disorder? And they were asking about 2 phenotypically normal couple for an autosomal recessive disorder. Would that also show 2 peak? Yes. So you understand how to read them, why they have a 2 peak, what's the, for sure you need the basis of the disease, the presentation and inheritance. When you are given a single gel, quickly decode it. You know very well from bottom to top, it's 5' to 3', not the other way round, right? Sorry, bottom to top, 5' to 3'. Yes, when you have 2 gel of newly synthesized sequence, as you see here, normal versus patient, but first, 2 things. When you are given a single gel, when you are given a 2 gel. When you are given a single gel, could quickly decode it from bottom to top, 5' to 3'. But since there are 2 gel, you can also compare triplets and save time. You know, this triplet change showed a change from normal to patient. The TGG changed to TGA. But tell me still, what are these two gel of? They are of still a, they are of what? Well, they are the sequence for a, what would you call them? They are the sequence for, these gel are the sequence for, you know, DNA sequencing. So this is it a newly synthesized strand? Is this a newly synthesized strand sequence? Yes, absolutely. So this is newly synthesized strand sequence. If one gel is given of the newly synthesized strand, first decoded. If there are two gel, please compare triplet to get to the change from normal to, from control to patient. Also, now please go as, please, when you want to do well in these exam 2 concepts, read as has been told. Now they are telling you, now they are telling you that the newly synthesized strand is the, what are they telling you? The newly synthesized strand that you decoded on the gel is the newly synthesized strand. Here is the sense strand. Absolutely. So if you want to know the change, you know the change was TGG to TGA. What is the relationship of mRNA to the sense strand? Is mRNA same as sense strand? mRNA sequence, is it same? Except instead of T, there is, instead of T, you have U. And read in frame, it's usually read in frame. Read in frame means you start from the, you start from the first nucleotide. So are you clear? So you're just following what is on the question. Let's see if this other question concepts are the same. So see, there were different question concepts. First question we did, they had a newly synthesized strand and they were just asking what? In the first question, there was a newly synthesized strand and they were asking what? Of it. First question, not this question. In the question that we just had. In the first question, there was a newly synthesized strand and a chromatogram. They were just asking template of it, template of the newly synthesized strand, right? Also, as I've always told you, whenever you want to, whenever they ask complementary, first find out complementary, then read from 5 prime. So in the initial question, there was a newly synthesized strand and they just ask you sequence of the template. But now look at the next concept. We already established in this one. In this one, what was the change? They gave you the newly synthesized sequence as you see in these two gels, normal versus control versus patient. But now they ask you to assume that the newly synthesized strand is the sense strand. So whatever you are being asked, you are doing, not putting your thought. Is that clear to you, right? Initially, they asked you, this is a newly synthesized strand. What is the template sequence of it? What is the sequence of the template or the strand that acted as a template for it? Here they are saying this is the newly synthesized strand, but now they're asking you to assume that this is the sense strand. So all I want to tell you is the first question concept that we discussed and this question concept same? Are they similar questions? Yes, is the first question concept same as the second question concept? The technique is the same, right? So do you understand the difference? Anyone? Are you understanding what I'm saying? Yes? Yes, right? So yes, the gel sequence is the newly synthesized strand, but then whatever is asked, are they asking what is the sequence of the strand that acted as the template? Or they're asking here to you assume that this is the sense strand. We go as the question goes. Now look at this. What is the sequence on the gel that you will decode from 5 prime to 3 prime? What is the sequence? This is the sequence of, I hope you know how to decode it, right? Yes, you know how to decode it. You move from bottom to top and start reading the nucleotide T, C, right? Does that make sense? But what is this gel sequence of? This is the sequence of from 5 prime to 3 prime, bottom to top. This is the sequence of, yes, anyone? This is the sequence of, do you see my cursor? No, not the coding sequence. That would be a wrong answer, please. You can still correct yourself. This is not the sequence, coding sequence. This is the sequence of newly synthesized strand. Whatever is given to you is a newly synthesized strand. Then they are asking you, like in the first question concept, what is the sequence of the of its template or the strand that acted as a template? Please, this is not the coding strand. This is not the coding sequence. This is the newly synthesized sequence. And first they are asking you, what is the sequence of the strand that acted as a template? So what will you do next? This is your newly synthesized strand, and they ask you, what is the sequence of the strand that acted as a template? What would you do first? What would you do first? First, complementary. Very good. Just find out complementary. Now, imagine, now they have asked you to assume that this strand that acted as a template is the sense strand. Now, this is the coding or sense strand. And sorry, you know that mRNA is? You know that mRNA is same strand. So now they're asking you to assume that this is the sense strand. The strand that acted as a template you just found out is the sense strand. Then you will find out mRNA sequence. Is mRNA same as sense strand, since you are assuming that this is the sense strand? Is mRNA same as sense strand? Instead of T, what would you say? Instead of T, it is U. And then read from 5 prime. Don't forget to read from 5 prime. Remember, mRNA is 5 prime to 3 prime, and then you decode the amino acid. So see, however the question concept or the technique concept presents. Are they giving you a newly synthesized strand and asking you template of it? Are they giving you newly synthesized strand and asking to assume that this newly synthesized strand is a sense strand? Or are they giving a newly synthesized strand, first asking you to find a template of the strand, and then asking you to assume that the template sequence is the sense sequence. But you will always keep that doesn't change. mRNA is same as sequence is same as sense strand. mRNA sequence is complementary to template, except instead of T, there is U. What is about microarray? How do you have two microarrays, DNA microarray and cDNA microarray. Which one is assessing upregulation and downregulation of gene expression? DNA microarray or cDNA microarray? cDNA microarray, which is assessing gene deleted or amplified, DNA microarray or cDNA microarray? DNA microarray. Which genes are deleted? Tumor suppressor genes. Which genes are amplified? Proto-oncogenes in the cancer. What you need to do is find out the color of what? Find out the color of, see my cursor? First and foremost thing is find out the color of control. What's the color of control here? What's the color of, sorry, give me a second. What is the color of control here? Give me a second because I want to see the, what is the color of the control here? Red. And what are they asking? Look at my cursor. What are they asking? Color of the control is red, and what are they asking? Look at my cursor. Color of the control is deleted. Give me your numbers. Color of the control. First, see the color of the control. And they are asking deleted. Give me your numbers. Deleted. Color of the control, genes deleted, 2, 6. Absolutely wonderful. I will explain you the principle in the end, but my concept here is to do a quick recap, then explaining you the principle. I've already told you initially in my lectures reviews or recaps. What is the color of the control again? Color of the control? Color of the control here is red. And what are they asking? Genes? What are they asking? Gene amplified. Give me the color numbers, please. Of gene amplified, 3 and 5. Absolutely wonderful. Let's go further. Again, if you see, can you tell me what was the color of the control? Because the green is amplified, genes amplified, and red is gene deleted. Color of the control was red. So your first and foremost aim should be find out the color of the control. What about this? Okay, when you go back, is this DNA, are they finding gene deleted or amplified or gene expression here? What is the interpretation here? Gene deleted or amplified, or gene deleted or amplified? Both, deleted and amplified. Though this is comparative genome hybridization, it's not DNA microarray, but has the same principle as DNA microarray. I've just brought in different slides just to tell you, okay, what do you think is the color of the control? Now, is it DNA microarray or cDNA microarray? Is it DNA microarray or cDNA microarray when you see expression? Is it DNA microarray or cDNA microarray? It's a cDNA microarray. Now you know that the down regulation is green. So what was the color of control? Color of control here was down regulation is green, please. Color of control was green. Absolutely, absolutely. Does that make sense? So that is why upregulation, color of the color of the control here was green, because down regulation is showing green. Do you understand that? Somebody said red. Can you correct yourself there? Remember, first you see color of the control, then deleted, amplified, down regulation or up regulation. Definitely here color of the control was green. That's why down regulation is, down regulation what binds? What binds with down regulation? Which color? Color of the control was green. Green, absolutely. That doesn't change. Is this DNA microarray or cDNA microarray? Is it DNA microarray or cDNA microarray? This one. So what is it assessing then? Upregulation, downregulation of gene expression. So you have a technique, they have a principle, they have an interpretation, and that is what you are reading in these pattern. So what is the color of control here now? Just a second. Okay, what is the color of the control here now? Green or red? Green. And what are they asking? Upregulated or downregulated? Downregulated. So give me the color. Give me your answer, sorry. Color of the control is green, then they are asking downregulated. Your answer is 5. Absolutely wonderful. This one trips me due to the... No, I think not exposed is green, a control. Not exposed is control. There's no difference in wording. Not exposed means this is the control. Is that clear now? Yes? Yes, I can give you questions if I explain you more when, let me finish it so that quickly boom, 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 and then I'll stop recording and we can have some time for question. Not exposed is control. And they're asking down-regulated. Yes, absolutely wonderful. 5 is the answer, right? Let's go to the next one. Sorry. Let's go to the next one. Which is the color of the control here? Green or red? Color of the control, green or red? What is the color of control here? Green. And what are they asking? So this is important. What is the color of the control and what is being asked? What are they asking? Color of the control is green and upregulated. So what is your answer? What would be your answer? 1 and 3, absolutely wonderful. Again, color of the control here. Color of the control. Color of the control here, green. And tell me if it is, now is it DNA microarray or cDNA microarray? Is it DNA microarray or cDNA microarray? What microarray is this? DNA microarray. So is it testing upregulation, downregulation, gene expression, or gene deleted, amplified? DNA microarray, amplified, deleted. And what did you say? Color of the control? Green. Tell me gene deleted and answer. Gene deleted in tumor and answer. What would be your answer? Color of the control is green. 3, 5, and 10. Let's see, is that okay? Look at that. 3, 5, 10. Yes? Absolutely wonderful. Let's go further. I'm trying to do a quick recap. Again, color of the control here is, color of the control here is, color of the control is very important. Not every time it's red or green. What's the color of control here? Red. What is the, sorry, what's the, I'm being too quick, but since it's a recap. So color of the control here is red. And tell me, deleted. Oh, sorry. This is, first of all, Color of the control. Color of the control here is red. Then is it DNA microarray or cDNA microarray? Which one is this? Is it a DNA microarray, cDNA or microarray? So what are you assessing? Gene deleted, amplified, or upregulation or downregulation? Upregulation and downregulation. So color of the control is red. So tell me downregulated. Downregulated. Color of the control is red, you said. And now tell me downregulated. Give me the number 3, 4, 9, 12. Let's see. Downregulated, right? You see my cursor here? 3, 4, 9, 12. Equal copies, you know, yellow. Not expressed, black. Equal copies, yellow. Not expressed, black. You're very clear with dicer being used. siRNA, miRNA. Which is more specific? siRNA. What do they target? Do they target replication? Do they target transcription? Or do they target post-transcript mRNA? What do they target? The post-transcript mRNA and interfere with the function, reducing what? Protein product. Right? Are you clear with that? Yes, you talked about miRNA, siRNA, dicer, but remember that they are not affecting at the miRNA, do not interfere with the replication, transcription, but post-transcript, they are targeting mRNA and reducing what? Production of a protein. So you should be clear with the principle. As I told you, there is whole genome versus whole exome sequencing. Where are you only detecting exons? Whole genome or whole exome? Only exons. Only exons is whole exome sequencing. That's why, what is the advantage? You are doing only exons. What is the disadvantage? Well, you might miss a mutation in the non-coding region, promoter region, introns. So that is why each technique has advantage and disadvantage. Which technique are they asking here? Whole exomes or whole genome? They are asking whole exome here or whole genome? Which technique is being whole genome? But Dr. Henshaw has clearly given you advantages, disadvantages of whole genome versus whole exome sequencing. How are you approaching population genetics here? I'm being quick so that I can address the key concept. First of all, you are aware of all these three terms, p square, 2pq, and q square. Which one is homozygous for autosomal recessive? Homozygous for autosomal recessive. Q squared, you know that. So these terms don't change, except when it comes to the inheritance pattern. Either you know the inheritance pattern, clinical presentation, through clinical presentation, if it is given, what is the inheritance pattern, or through disease. So that is first key step. What are the steps? But before we go to the step, you should be clear with these. What do they reflect? And p squared, that's why here clearly is saying homozygous for autosomal dominant. Very good. When I ask you homozygous for autosomal recessive, you said Q square. Now let's see what is, how do you approach these concepts. First, inheritance pattern, then what is given, and then what is asked. Please, please, and please, as I told you, what is the disease incidence or disease frequency for autosomal recessive? Is it Q squared or Q? Disease frequency or disease incidence for autosomal recessive is Q squared. So Q squared is 36%. And what are they asking? So what is the inheritance pattern first? What is given and what is asked? So very good. What is the disease incidence or disease frequency for autosomal recessive is Q squared, and they are asking Q. Very good. What changes here in the next question? Same autosomal recessive disease, same Q squared, but now what are they asking? Instead of Q, what are they asking here now? Instead of Q? So what is the inheritance pattern? What is given and what is asked? What are they asking here instead of Q now? Anyone? Yes, this question? They are asking P. What is P now? How do you calculate P? P is 1 minus Q. So that is the change. See that what is carefully, what is the inheritance pattern? What is given? What is asked? What is this inheritance pattern now? Sickle cell. Sickle cell, what's the inheritance pattern here? Sickle cell. Sickle cell is autosomal recessive. What is this? Disease incidence, disease frequency. What is this? This is 1 by 400 is 1 by 400 is 1 by 400 is Q squared. And what are they asking? Carrier frequency means 2pq. So you understand that what is the inheritance pattern, what is given, and what is asked. Disease incidence for autosomal recessive is Q squared. But what are the inheritance pattern here now? How has it changed? What's the inheritance pattern here? It is autosomal dominant. What are the possible phenotype for autosomal dominant? What is the mutant allele in autosomal dominant, first of all? The mutant allele for autosomal dominant is little a or capital A? Capital A. What are the possible genotype of autosomal dominant phenotype? What are the possible genotype of autosomal capital A, little a, and capital A, capital A? Capital A do not survive. And that's why for autosomal dominant, the incidence of disease or frequency of disease is not p squared. What it is? What is it? The incidence of disease here is the 1 over the incidence of disease, which is, give me a second, what is 1 over 5,000 is what? 2pq. And what are they asking you? What are they asking you here? P or Q? P or Q? P or Q? What are they asking you? P. Does that make sense? So yes, this is different. This is a different inheritance pattern. It is autosomal dominant, Marfan syndrome. Capital A do not survive. So here disease incidence becomes disease incidence or disease frequency becomes 2pq, and they're asking you p. So whatever is given and whatever is asked is different for different question concept. Now what is here? So I'm just bringing your attention to that rather than teaching you all. So let's see that again. What's the inheritance pattern here? The inheritance pattern is autosomal recessive. So what is 1 over 3600 here? Q squared or Q? It's Q squared. Incidence of disease or disease frequency is Q squared. But they are clearly saying that Tony is an unaffected sibling. So in autosomal recessive disorder, when both parents are carrier and the sibling they are asking is unaffected, do you bring in 2/3 rule? Does 2/3 rule comes in? Is the carrier probability 2/3? So carrier probability 2/3 is of dad or mom? Dad or mom here? Dad or mom? Dad. Right? How will you find out the carrier probability of mom? From population genetics. So what is important for this question? How is it different? First of all, whenever Q squared is given or disease incidence is given, disease frequency is given, quickly find out 2pq, because you will need carrier probability to be calculated to find out the probability child is affected. Now, are there two phenotypically normal couple for autosomal recessive disorder? Is this a mating between two phenotypically normal couple for autosomal recessive disorder? Are both parents carriers? Are both parents carriers? When both parents are carriers, what is the chance or probability a child is affected in an autosomal recessive disorder? 1 by 4. So probability dad is a carrier, probability mom is a carrier times child is affected. That is how you will know the chance that they will have an autosomal recessive disorder. That's how you will know, which is asked in the question, right? They're asking you the chance that Tony and Maria's child would be affected by an autosomal recessive disorder. So you understand this is different. This is autosomal recessive disorder. Here 2/3 rule comes. Here they are two phenotypically normal couple and they are asking you the chance the child will be affected. Now what is different here? This is this autosomal recessive disorder? Is this autosomal recessive disorder? Oh, 1 by 30 was derived from population genetics. If you see here, so because somebody has a quick question, 1 by 30, because both, so in autosomal recessive disorders, if they are asking you the probability child is affected, should both parent have the mutant allele? Yes. So this is the mating between two phenotypically normal couple because they are not affected, right? Somebody asked me, where do I get 1 by 30? You know carrier probability of dad because of 2/3 rule. Do you know carrier probability of mom? Did you know carrier probability of mom? No. How did you find it out from? The disease incidence in the population. So this is where you calculated the 2PQ. Somebody is asking me, are you clear now so I can move on, please? Whose question was that? Where did 130 come from? Yes, 130 came from the population genetics, the incidence of the disease. This is how you calculate it. So last question was autosomal recessive disorder, two phenotypically normal couple. That means both have to be carrier and they are asking child affected. Now, is this autosomal recessive disorder? Or something has changed? Is it autosomal recessive disorder or something has changed? It's an X-linked recessive disorder. Now here disease incidence and disease frequency is Q squared or Q? Q squared or Q? Q, please know that. Quickly find out 2PQ. When you say mating between two phenotypically normal couple in X-linked recessive disorder, who's the carrier, mom or dad? Who's the carrier, mom or dad? Mom is the carrier. So do you want to find out carrier probability of mom from population genetics? Do you want to find out carrier probability of mom for population genetics? No, no, no. It's always like that. Now, males are not carrier, please. It's not about male and female. In X-linked recessive disorder, incidence is Q. It's not sex related, right? It's not about sex. It's not like male or female. Does that make sense? I will answer your question, Samir. No. No, it's not about male and female because you only need one mutant X to express the phenotype. That is why the disease frequency or disease incidence for X-linked recessive is Q. It's not about male or female. Yes, moms and daughters are carrier in X-linked recessive disorder. So this is if this is the mating between two phenotypically normal couple in X-linked recessive disorder, I want to go back. Who's the carrier? If it's a mating between two phenotypically normal couple, as they are saying in X-linked recessive disorder, who's the carrier here? Who's the carrier here? Who's the carrier here is mom. So do you want to calculate carrier probability of mom? Yes. And if there's a mating between two phenotypically normal couple in an X-linked recessive disorder where mom is a carrier, what is the chance child is affected? Children are affected. If it's a mom is a carrier and dad is unaffected, 1 by 4. So two phenotypically normal couple, X-linked recessive disorder, naturally mom is the carrier, mating between them two, the child affected is 1 by 4. So why are we doing carrier probability only of mom and not dad? Why are we doing carrier probability only of mom here? Because dad is not a carrier. They are two phenotypically normal couple in X-linked recessive disorder. Mom and daughters are carrier. And also, please, as somebody asked me, irrespective of male and female, disease incidence and disease frequency for X-linked recessive is Q. It is not dependent on the sex. Right. So how did we do these problem set? We first identified the inheritance pattern. Then we see what is given. What is given? So what is given here? What is this 9%? What is 9%? Is it Q squared or Q? It's Q. And what are they asking? What are they asking? Naturally, what are they asking here? Q squared. And what are they asking? What are they asking? 2PQ. So what is the inheritance pattern? What is given and what is being asked? Disease incidence and disease frequency for autosomal recessive is Q squared. For X-linked recessive is Q. And for autosomal dominant is disease incidence and disease frequency for autosomal dominant is what? Autosomal dominant is disease incidence and disease frequency for autosomal dominant is 2PQ. Disorder again, either if it is given by the disease or by presentation. The only thing here changes, which is an exception. So what is the disease here again? Autosomal recessive. And why are we doing all this? Remember, this is all population genetics is based on two phenotypically normal couple. If it's for autosomal recessive disorder, they have more chance to have a disease. So they want to come for counseling. and find out what is the probability a child would be affected, right? It's a part of also counseling, genetic counseling. So what is the disorder here? Autosomal recessive. This is a little glitch here. Here they have not given you disease incidence or disease frequency, but what have they given you? This is a exception. This is autosomal recessive disorder. Instead of disease frequency and disease, they have given you gene or allele frequency, and you quickly find out 2PQ, which they are asking. This is just an exception. Autosomal recessive, they have given you allele frequency or gene frequency, but otherwise, what is the disease frequency or disease incidence for autosomal recessive disorder? It is, for autosomal recessive disorder, it is Q squared. Again, autosomal recessive disorder. I think I want to tell you one thing, but anyways, I just want to do a quick recap. If I remembered, I want to just go back. Okay, yes, the one point I wanted to tell you is all enzyme deficiency, most of them are autosomal recessive unless specified. Do you see albinism? What is albinism? Albinism. Autosomal? Albinism. All enzyme deficiencies are autosomal recessive unless most of them is autosomal recessive. Also, you can be aware of presentation in albinism. What will you see? What will you see in albinism? Lack of what? Sorry. Albinism is lack of what? You should be familiar with presentation, especially which are given in the lecture slides. So albinism is lack of what? Pigmentation of skin color. Absolutely. You will not see that in cystic fibrosis, right? A lack of pigmentation you will see in albinism. So also presentation, again, autosomal recessive, Q squared. They are asking 2PQ, but what have they added? One more layer. It's times. What have they added? One more layer is times 26,000. So see what is the inheritance pattern, what is given, what is asked, and don't miss any step. That is different here. What is difference in the next? It is again autosomal recessive. What is 9% here? What is disease incidence or disease frequency for autosomal recessive? What is the disease incidence and disease frequency for autosomal recessive? Q squared. What are they asking? Phenotypically normal. Now, phenotypically normal for autosomal recessive as well as X-linked recessive are. Who are phenotypically normal for autosomal recessive and X-linked recessive? Carriers. And who are phenotypically normal? If it is, I'm not asking genotype, I'm just asking in general. Who's phenotypically normal for autosomal recessive as well as X-linked recessive? Who are phenotypically normals? Carriers and normal, right? Yes, you're right in saying carriers and normal. You're right in saying you can also address the genotype, carrier and normal. Yes, but somebody's saying in X-linked recessive, carriers are mom or daughters. That is specific for X-linked dissociative disorder. But what is phenotypically normal for both autosomal recessive and X-linked recessive is carrier and normal. Do you understand the general concept which I was asking? Are you clear with that? You understand my point? Yes. But yes, you can also tell me the genotype. So they are asking both the carriers and the normal for autosomal in case of autosomal recessive disorder. But they have also added one more layer, which is 36,000. So there are layers. They are differently asked. But first step is inheritance pattern. Next step is disease incidence. Third step is quickly find out 2PQ. But then don't forget what is asked. Cystic fibrosis, autosomal recessive. 1 by 2500 is the disease incidence, Q squared. Here it has just changed. This is not a mating. Is this a mating between two phenotypically normal couple? Is this a mating between two phenotypically normal couples? So what has changed here? No, this is the mating between an affected woman for autosomal recessive disorder and an unaffected man. Here, is dad a carrier? Is the dad a carrier here? Is dad a carrier here? It's autosomal recessive. Mom is affected. Is dad a carrier here? Absolutely. Otherwise, you cannot have a phenotypically a child with a autosomal recessive phenotype. But is it possible in X-linked recessive disorder that only one parent is carrier? Is it possible in X-linked recessive disorder that only one parent is carrier, and who's the carrier? Mom or dad? Mom, right? So here what will change is the probability child would be affected. This is not between two phenotypically normal couple for autosomal recessive. This is between affected mom and carrier dad. So here probability is what? It's not 1 by 4. The probability here is child is affected instead of 1 by 4. Half, yes. Again, albinism, X-linked recessive, they have given you Q squared. They are asking you, they have clearly given you that the sibling they are asking is unaffected. What comes here? When there is a sibling which is unaffected, any rule comes here? Yeah, if mom is not affected, yes. If this is between two phenotypically normal couple for autosomal recessive disease, child affected will be 1 by 4. Yes, mom here has a 2/3 rule. Somebody is asking a question that if it's an autosomal recessive disease and it's two phenotypically normal couple and mating, what's the chance the child will be affected? Yes, then it will be 1 by 4. Yes. Are you clear with that? Did I answer you? Yes. Here, 2/3 rule is applying to mom. And how will you find out carrier probability of dad since it's autosomal recessive? Should dad be a carrier too? Since it's autosomal recessive, should dad be a carrier too? Yes, because both are phenotypically normal. You know this is 2/3. Well, how will you find the carrier probability of dad now from? So this is the mating between two phenotypically normal couple for autosomal recessive disorder. 2/3 rule applies because this is an unaffected sibling. So how will you find out carrier probability of dad here? From where? From population genetics, 2PQ, and probability when two phenotypically normal couple or carrier is mating in autosomal recessive disorder is 1 by 4. Does that make sense? So you should be careful who has the 2/3 rule. The other person, the other parent, you have to find out the carrier probability from population genetics. So is this a mating between two phenotypically normal couple? For autosomal recessive disorder, was the last question a mating between two phenotypically normal couple? For an autosomal recessive disorder? Yes, everyone? Are you clear? Yes, but this is it autosomal recessive? Have they given you Q squared? What do you do? Oh, sorry, now, just a second. Sorry, I would be, just as an exception. Is albinism autosomal recessive? I told you enzyme deficiencies are autosomal recessive unless specified, most of them, right? Sorry. So is this autosomal recessive? But here, see, have they given you disease frequency or disease incidence or gene frequency or allele frequency? So here this becomes Q squared or Q? This becomes Q, that's all. Otherwise, disease incidence, disease frequency of autosomal recessive is Q squared, but they are giving you gene frequency, allele frequency. So that's why just an exception. This is not normal. This is exception for autosomal recessive that you will assume it Q. And is this a mating between two phenotypically normal couple? No. This is the mating between? Is this a mating between two phenotypically normal couple for autosomal recessive? What is this mating between? An affected mom and this is the mating between an affected mom for autosomal recessive and a carrier dad. Absolutely wonderful. Unless they tell you here for autosomal recessive that is a gene or allele frequency. Otherwise, disease incidence and disease frequency is Q squared, please. Again, autosomal recessive, enzyme deficiencies are autosomal recessive, most of them, unless specified. This is Q squared. They're asking 2PQ. Very simple, right? This is Duchenne muscular dystrophy. What is the in the first step? Inheritance pattern. Then what is given? Then what is asked? So tell me, what is this inheritance pattern? X-linked recessive. What is 2.8 over 100,000? What is this? Is it Q squared or Q? Is this 2.8 over 100? It is Q. You quickly find out 2PQ. Now they are saying this is a mating for X-linked recessive disorder between two phenotypically normal couple. So who is the carrier here in X-linked recessive disorder? Who's the carrier? Mom or dad? Mom. So this is the carrier frequency. This 5.6 over 100,000 is a carrier frequency of what? This 5.6 over 100,000 is the carrier frequency of mom or a dad? Mom or a dad? from population genetics, mum. And when two phenotypically normal couple is mating in an X-linked recessive disorder, what is the probability child is affected? What is the probability child is affected? 1/4. Please do Punnett square, not pedigree. Please do Punnett square, right? What was for epigenetics? Epigenetics were very simple. Two modification, histone versus DNA. Histone are the histone modification is common, acetylation, deacetylation. Are they epigenetic modification? Absolutely. Acetylation leads to euchromatin or heterochromatin. Acetylation leads to transcriptionally active euchromatin or transcriptionally inactive heterochromatin. Acetylation leads to transcriptionally active euchromatin. So what is the modification? Where it is happening? Histone acetylation will lead to transcriptionally active euchromatin. Then was there another modification? Modification of DNA. Modification of DNA was methylation, and that led to modification of cytosine of DNA was methylation leading to silencing. Is it a normal phenomena? Yes. This is because those genes are silenced, those traits will not be expressed. It's a normal phenomena. Do you recruit MECP2 protein when you are modifying cytosine of DNA? Is this protein recruited in the process of silencing or the outcome of silencing? Yes, this is recruited. And are there two clinical disorders? We have talked about that, Fragile X and Rett syndrome. Both of them is about cytosine methylation, silencing, but which one is about CGG repeats towards the 5' end, cytosine silence, frataxin protein reduced. Frataxin protein reduced. It is Fragile X or Rett syndrome. Fragile X and Rett syndrome both are X-linked dominant. So there are two different disorders affecting having two different etiopathogenesis. CGG repeat is not Rett syndrome. CGG repeat is Fragile X. Also, this is a normal phenomena. CPG islands means on the same strand towards the 5'. This is a normal modification where cytosines of DNA are modified, leading to gene silencing. There is a cascade. We will bring that too. So, and when that happens, that trait will not be expressed. But this is the normal phenomena. What happens in Fragile X? Since you have a CGG repeat, there also cytosine is silenced, right? In Fragile X, you have CGG repeats, right? Yes or no? Yes, they are pathological trinucleotide repeats. In Fragile X, you have a lot of CGG repeats where cytosine is silenced. But this CPG is a normal CPG islands on the same strand near the promoter, which are modified by methylation leading to silencing, right? Why is this natural phenomena? Naturally, if those genes are silent, that trait will not be expressed. So are you clear with this silencing cascade? Yes. Does this silencing cascade involve MECP2? Does this silencing cascade involve MECP2? Has MECP2 anything to do with Fragile X syndrome? Is MECP2 defect leading to Fragile X syndrome? No. So the Fragile X and Rett syndrome, which are X-linked dominant, disorder, have different etiopathogenesis. Now, is this about Fragile X or Rett syndrome? Yes. MECP2 defect is not Fragile X. Fragile X is CGG repeats, near 5', cytosine methylated, promoter silenced, and frataxin protein low. Yes, MECP2 is not, defect is not Fragile X, right? Here, are they talking about Fragile X? And when you look at the question stem, or Rett syndrome? Are they talking about Fragile X or Rett syndrome? Is it about Fragile X or Rett syndrome? Rett syndrome. So don't mix and mess them up. X inactivation is random, 50-50 usually, but sometimes it's skewed. Where is it skewed? Then it's called non-random. It's skewed towards the mutant X. And what does that result in? A number of possible phenomena, but for X-linked recessive disorder, it may result in manifesting heterozygote. For X-linked dominant disorder, the skewed X inactivation, which is towards the mutant X, may lead to no phenotype in the mom, but daughter may have the phenotype. So yes, skewed X inactivation is non-random, usually towards the mutant X. Is Duchenne's X-linked recessive disorder? Is Duchenne's X-linked recessive disorder? Yes, are carriers symptomatic here? Are carriers symptomatic in X-linked recessive disorder? Are carriers symptomatic in X-linked recessive disorder? No. Are carriers mom here? Are carriers moms and daughters here? Yes. But this is saying that the carriers are symptomatic, which is not a normal phenomena. So are you aware of random and non-random X inactivation? Yes, non-random is skewed X inactivation, skewed towards the mutant X. Do you want to understand this normal phenomena of methylation of cytosine and gene silencing with Prader-Willi and Angelman? I will explain you just Prader-Willi. You can apply that to Angelman. Which one has obesity as a clinical presentation? Some clinical presentation you should be aware of. As I told you, lack of pigmentation is not cystic fibrosis, right? Lack of pigmentation is which disorder? Lack of skin pigmentation is albinism. So some clear clinical presentation you can also be, relate from your fall. Prader-Willi and Angelman, both 15 chromosome, different gene. So in Prader-Willi, what is intact, paternal or maternal? Genes intact, paternal or maternal? Paternal. There are only 3 mechanisms that lead to Prader-Willi syndrome: methylation, deletion, and uniparental disomy. So if Prader-Willi paternal genes are intact, and remember, you have to knock out both the genes. So deletion and methylation should be paternal or maternal? Paternal. And uniparental disomy should be in Prader-Willi maternal, because maternal. And which has obesity? Prader-Willi. See, methylation and deletion for Prader-Willi is, methylation and deletion for Prader-Willi should be, methylation and deletion would be paternal or maternal? Prader-Willi. Methylation, paternal, leading to. So this is the cause leading to Prader-Willi syndrome. Same chromosome, different genes. Prader-Willi is obesity. Please, not obesity is Prader-Willi, not Angelman syndrome. What is intact in Angelman genes? Maternal. You do the same 3 mechanisms: deletion, methylation, and uniparental disomy. They are clearly telling you Prader-Willi here. They are telling you there is no deletion or methylation. So both copies in Prader-Willi should be coming from what? Origin should be for Prader-Willi. Both copies, uniparental disomy. For Prader-Willi, maternal. So he understand that they are same chromosome, different genes. Now this should not be a heavy segment for you, though drugs are heavy, right? But you have to do 4 deconjugating enzyme, as Dr. Wang has said. Dr. Wang has really beautiful, great lectures. You have to do CYP2D6, CYP2D6, methyltransferase and acetylase. First step is to identify the deconjugating enzyme. CYP2D6, CYP2C9, whether it's CYP2D6, it's CYP2C9. Is it acetylase or is it methyltransferase? Is it acetyltransferase or methyltransferase? After you identify these drugs, please know whether, after you identify which is the deconjugating enzyme, come to the drug. Are they asking about the drug and what does this enzyme do to the drug? Is it converting pro into active, active into inactive? Then third step, are they concerned with ultra rapid metabolizer or poor metabolizer? There are only 2 red flags: or increased activity, decreased activity. Then due to this isoform, what is the outcome? Is it lack of efficacy or is it side effect? So again, I'm repeating the steps. First, you have to identify what are the deconjugating enzyme. And then I'll clearly explain you in this slide why knowing those steps are important, right? First, you identify which is the deconjugating enzyme. Is it CYP2C9? Is it CYP2D6? Is it acetyl transferase or is it methyl transferase? Then you identify the drug. Is this enzyme converting pro into active or active into inactive? Then which isoform is being investigated? Ultra rapid, rapid metabolizer, poor metabolizer, increase or decrease activity. Once you have done that, what is the outcome? Is the outcome of that isoform side effect or lack of efficacy? Why? Because the physician has to sorry, just blabbering. Sorry, because physician has to adjust the dose maybe, or physician has to see maybe reduce the dose. A physician has to see is there will be effective treatment. So that is why first first detoxifying enzyme, is it CYP2D6, CYP2C9, acetyl transferase, methyl transferase. Then what are they doing to this drug? Pro into active or active into inactive. Then what is the isoform in question? Ultra rapid, poor metabolizer, or simply increase and decrease activity? Either one could be there. And this isoform is leading to what? Lack of efficacy of the drug or too much side effect of the drug, because physician has to change the treatment plan. For example, when you compared, this is the same enzyme, CYP2D6 here, CYP2D6 here. Tell me, ultra rapid metabolizer will lead to a lack of efficacy in which one? Codeine or nortriptyline? Ultra rapid metabolizer will lead to lack of efficacy in case of codeine or nortriptyline. Ultra rapid metabolizer will lead to lack of efficacy of drug in nortriptyline, which is written here, and it's not the same as this. So that is why it's important for you to have which detoxifying enzyme and what is it doing to it. Absolutely wonderful. Ultra rapid metabolizer in case of nortriptyline will lead to lack of efficacy, not side effect. Is that clear? Right? So you should know that information, and it's clearly given in this one. Now tell me, as I asked you, in case of codeine or nortriptyline, which isoform will lead to side effect? Which isoform, okay, I should ask you a different way. In codeine or nortriptyline, where ultra rapid metabolizer will lead to side effect? Where ultra rapid metabolizer will lead to side effect in case of codeine or nortriptyline? Codeine. Then you should know side effect of morphine accumulation also. What is side effect of morphine? Respiratory depression. So you should be familiar with side effect too. When you are saying, as I asked you, codeine or nortriptyline, where ultra rapid metabolizer will lead to side effect? Very correct answer. Absolutely wonderful. Codeine. But then what is the side effect? Respiratory depression. And Dr. Wang has clearly stated side effect if there are side effects due to that isoform. So please see that isoform is the cause, or that isoform leads to lack of efficacy or side effect, not the other way around, right? Very wonderful answer. When I ask you ultra rapid metabolizer in this case, which isoform will lead to side effect? Codeine. Ultra rapid metabolizer in case of codeine, CYP2D6 ultra rapid metabolizer will lead to side effect, which is respiratory depression, right? Here, which is the enzyme? Tell me. First, tell me what is the detoxification enzyme that we are talking about. The detoxification enzyme is CYP2D6, and drug is which one? The next step is drug. Drug is which one? So enzyme is tamoxifen. So what does CYP2D6 do to tamoxifen? Convert prodrug into active drug or active into inactive? Tamoxifen into endoxifen. Pro into active drug. So you went wrong. Don't go wrong in your steps. CYP2D6, tamoxifen into endoxifen, pro into active drug. So what will ultra rapid metabolizer do? Best answer is good. Best answer is good. It will respond, more active drug. So first, detoxifying enzyme. Here it is CYP2D6. Drug in question, tamoxifen. What is CYP2D6 doing to tamoxifen to endoxifen? Pro into active drug. So what will ultra rapid metabolizer do? Absolutely good. More more active drug and more response to treatment. As this is the best answer here, right? That it will respond well to the treatment. I want to just go back. No, it's okay. You can also correct yourself now. I'm explaining you the step. You still have a lot of time. I'm just being quick. So keep your, keep your spirits high, okay? You can still do it, okay? Great. So again, as I ask you here, first you know the enzyme, detoxifying enzyme, here is CYP2D6, then see what is the drug in question, and that's why I asked you one simple question. In case of codeine or nortriptyline, I'm asking you again, ultra-rapid metabolizer isoform will cause side effect in codeine or nortriptyline, and ultra-rapid metabolizer will cause side effect in case of an ultra-rapid metabolizer of CYP2D6 will cause side effect in case of codeine or nortriptyline. Codeine, yes. And then you should know the side effect. Also do only one way. Either know what will cause lack of efficacy or lack of side effect. You can, don't take both at the same time. This is my detoxifying enzyme. This is my drug. What is it converting into? Proactive, active, and inactive. And then just do one isoform. What will that lead to? Naturally, the other isoform will lead to the opposite. Again, here, CYP2D6, tamoxifen is in question. It converts pro into active drug. You should know the side effects of only the drugs which are in Dr. Wang's lecture slide. Only those which are in Dr. Wang's lecture slide, please. Yes. You should know, great. And here, because ultra-rapid metabolizer will lead to more active drug, well, good for the treatment. Here, simply, what is methyltransferase doing? Metabolizing 6-mercaptopurine. So tell me, if there is decreased activity of methyltransferase, what will that lead to? Methyltransferase is just metabolizing 6-mercaptopurine. So what if there is a decreased activity of methyltransferase, what will that lead to? 6-mercaptopurine toxicity. Just the drug name and the side effects too, yes. What is the drug, what side effect? And Dr. Wang has clearly given you that. So identifying the detoxifying enzyme, identifying the drug, is it converting pro into active, active into inactive? Then isoform, ultra-rapid, poor metabolizer, increase or decrease activity, what is it leading to? Side effect or lack of efficacy? Yes. Now, what is the drug in action here? Is it CYP2D6, CYP2C9, acetylase, acetyltransferase, or methyltransferase? What is the drug in question here? CYP2D6, CYP2C9, acetyltransferase, or methyltransferase? What is the drug in question here? Acetylase. Then, what is the drug? Is it converting this drug from pro to active or active into inactive? Pro to active or active into inactive? Pro to active or active into inactive? What do you see in the question stem? A lack of efficacy of drug, or due to the isoform, or too much side effect, too many side effects? Lack of efficacy of the drug, too many side effects. So which isoform will lead to too many side effects? Ultra-rapid metabolizer here or poor metabolizer here? Ultra-rapid, poor metabolizer. So do you see that you needed the information of what is the detoxifying enzyme, the drug, is it converting active into inactive, which is not in the question stem? Then only you could identify that ultra-rapid metabolizer will lead to tingling and sensation of isoniazid accumulation. So yes, those side effects you should know, and Dr. Wang has clearly given you in the lecture slide. I wish you good luck. Okay.
Thank you.
One day. Take care.